Working in the Department of Computational and Data Sciences, IISc Bangalore, and exploring the areas spanning genomics, proteomics, metabolomics, drug discovery, algorithms, and methods with a focus to address the biological problems with computational biology and bioinformatics. May we join hands to welcome Dr. Debnath Pal, Professor, to address the gathering.
Hello, everybody. I thank Dr. Mohan Kumar for inviting me to this symposium and giving me this opportunity to share my research with you. First, before I begin, a disclaimer: I'm not a crop scientist, so I will not be talking on crops, but whatever I'm going to talk are basic fundamentals of genomics and understanding biology in general. So the concepts I'm going to share with you would be equally applicable to crop science as well. And as I progress, I will try to point similarities, and you can try to, use those similarities and adapt the concepts to whatever domain you're working on. So let's begin. So this is not crop, this is human genome. But you will have a crop genome, maize genome is amazing, as you know. Arabidopsis is very well studied. But in our lab, we work on human genome. Okay? So human genome is very large. It has about 3 billion bases. And because it's very large, it poses what is called a data science problem. So you'd have to typically, analyze a lot of data. But what do you do with this data? We had speakers in the previous session reminding us that now we are doing the biogenome project where we'll have more and more data. So how are we going to analyze all these data? This is a big challenge for all of us. So human genomics typically asks questions on, how similar we are. So the same with plants, right? You do plant breeding, you try to understand, which crops are similar, and then you want to breed those. You want to cross those, and you want to get new traits out of them. So it's very similar to what you do in plants, and it's what we do in human genomics as well. And then we look at polymorphisms, and then we try to understand, what are the polymorphisms here, the linked SNPs, they don't affect the protein production. There are causative SNPs that are affecting the gene, and then you have to see which polymorphisms are missense and what affect the protein. So in the realm of all this analysis, our idea is to find out which changes are functional, right? If I may coin the word, the functional changes in the genome, okay, that alters the trait. Okay, so trait is also something that you want to know it in crop science, and this trait arises due to the alteration in the genome. So we want to know the same things in human genomics as well. And the main problem that we face, as in any other genome, including plant genome, is the large presence of repeat elements. Okay, and you have to deal with these repeat elements. Some repeat elements are 100%, same 70%, and you have to figure out, which repeat elements, are of similar functionality. They are doing similar kind of jobs, and which are not. And these are not very easy things to do. And this repeats in the genome are one of the major challenges in any genomic analysis, be it in plants or in human genome. Okay, so human genome, you may know, has 50% repeat elements in it. Okay, plants have more, so the problem is more acute in plants. So nowadays, when we try to do genomic analysis, we do what is called the genome resequencing pipeline. Okay, this is standard genomic analysis, approach that one uses, where you extract the genome, the genomic DNA, you sequence it, and then if you have a reference, you essentially try to align the reference. The reads that you get from your sample, from the sequencer, you try to align it to the reference, and whatever you cannot align, you try to assemble it, and then together you try to find out the polymorphisms, the copy number variants, the large structural variants, and so on and so forth. And then you try to see from the database if there is any existing information, if you can dig out some information out of it. Okay. So this is the standard genomic pipeline that we currently use. And when we talk of any genomics related study, this is where typically it ends, right? So you culminate your study by trying to confirm the variations that you see in the database and what they do, and you identify some novel variations, and you try to decipher what they do. Okay. Now, you must understand that the genomic information is a one-dimensional information. It's just a string of letters that represents a chromosome, right?